stop recording. Okay. It just so happens that today is actually quite an excellent day to be sick, because today we're going to be pretty much lecture focused. All right. So today we're talking about carbohydrates. Carbohydrates, no pun intended, are pretty cut and dry, or at least the way I teach them, they tend to be pretty cut and dry. It's good to read over the chapter, and there's a lot we could talk about, but we just probably won't have time about. If we do have any time at the end of class, I'd say we could take the opportunity to answer enzymes, but you guys know me, I love to talk, so we'll see if we have time. Last time I taught this, we didn't have time at the end of class, but we'll just have to wait and find out. I'm going to set myself my 35-minute timer now, so when my alarm goes off, we're going to take a brief break to get up, move around, go to the bathroom, etc., and then we'll start lecture back up again. Okay, so today again, we're talking about carbohydrates. Carbohydrates is the last chapter of content that you will be on the hook for exam 2. Does that mean that you should check out for nucleic acids? Absolutely not, because you aren't going to be quizzed on nucleic acids for another month. This is not because I wanted it to be that way. This is because of the way spring break falls and the way signaling, and we have a paper discussion that's based on protein structure and signaling. So I wanted to do our paper discussion right after signaling. I didn't want you to have a quiz and a signaling discussion on the same day. I wanted to keep them separate in terms of so you weren't focusing on two different assessments at the same time. So I separated them. So you've got it. Don't let it fade away. I'm sorry. I apologize for the really long gap, but sometimes with classes, you just have weird stuff happen like that because of the way the semester and the content fall. Okay, so yes, we'll be doing nucleic acid. We'll be doing nucleic acids. We'll be doing nucleic acid and nucleotides and nucleic acids on Thursday. So that's where you'll be going over the basics of what a nucleotide looks like. A lot of this should be review, but you'll get it in more detail, and you'll have a nice video with a PowerPoint to go back over. We will, for nucleic acids and nucleotides and then nucleic acid structure, on Monday, we will be reverting back to our old format where you'll be reading a book chapter, watching a video, we'll be doing problems in class. But that's going to be the last. So signaling is a bit of a hybrid, but that's going to be the last class where we're really going to be exclusively problem solving. So for those of you who weren't fans, or for those of you who favor lecture, this is excellent news for you. But throughout the rest of the term, I am committed to trying to have you interact with your groups like every 15 minutes or so. So I'll probably literally just copy and paste this block and then put it on the next PowerPoint. But there aren't any notes or anything due for today. Okay. Again, the take-home quiz is due Thursday. I'm just going to collect them at the beginning of class. If you have a paper copy, bring it in. If you'd prefer to email, that's fine. I'll probably print it out and grade it on paper, honestly. But whatever is your preference, that's fine. We have exam 2 coming up next Thursday. And in preparation for that exam, and also bearing in mind the quiz, we have 2 help sessions this Wednesday and next Wednesday. Don't like to have to do the same day twice in a row, but just based on availability of you guys as a group, and for the availability, I think I was able to accommodate everyone's schedule except 2 people who responded. But bearing in mind, I want everybody to have a chance to ask questions. So as always, I can meet during office hours after class. I'm happy to meet before class, although you do have the risk of us getting sidetracked a little bit sometimes, but before class definitely does work. And I'm open to appointments. Some of you have already taken advantage of that. However, another option, if you don't want to get up before class, you're busy after class, you don't want to come down to Wake Downtown for an appointment, you can also email me a question and just put in the subject that this is a question for the problem session. I can ask it during the problem session, and it will be in the recording. So you can get your question answered. I try to post the problem sessions then afterwards. So it'll be a situation where I'll try to have it producing in my car and then post it when I get home, or the morning of. But in this case, because it's Thursday, I can try to get it up as soon as I can after the problem session. But I'm happy to ask questions. So not ask and answer questions. And then lastly, thinking out even further, you're going to be getting a short paper to read over spring break. I mean, I guess you could also read it in the, Monday and Tuesday between spring break and the paper discussion, whatever you choose, but it will be posted to the Sakai site after exam 2 and between the start of spring break. And it is a short paper. I think it's a Nature paper, and it's on the structure of the mu opioid receptor. So it ties into the signaling section, but it ties into protein structure and function. It ties into protein ligand binding, but it also ties into the dope-sick reflections. So I've gotten dope-sick reflections. I haven't physically counted them, but I'm pretty sure I've already turned them in. And I've really enjoyed, I haven't graded them yet, but I've enjoyed my preliminary skim over them so far. It seems like the book has really hit home for a few of you, for many of you guys, or changed your perspectives on different things. And I'm glad that you seem to enjoy it so far. Again, for this book, you're only required to read chapters, I think, 1 to 4 and 10 to 13. A lot of the stuff between those chapters is more personal anecdotes about addicts and their families, and about the structure of essentially the drug ring in southwestern Virginia. You're welcome to read that on your own time. I highly encourage you to read it, but you're not required to read it. I'm not going to force you to read that part. I'm mainly focused for this part. It was learning about how the epidemic started and how were the scientists and the doctors and the business people culpable, at least in some part, so partly culpable, how addiction as a disease might look different than what we thought it did before reading this book. I know it did to me. And also the last 3 chapters I'm having you guys read, I think 3, it talks more about medication-assisted therapy, which is as well as traditional abstinence therapy, and kind of how the science behind the science that can be used to treat addiction. So I want you to read about that, and we'll also be talking, although it won't line up with the book, we will be talking a bit about that during our discussion for the paper, and perhaps our discussion with the paper will help you as you're reading and giving a critical eye to the last portion of the book. If anyone is interested, is anybody here a fan of NPR? Am I the only NPR nerd in the room? Okay, we have another or two. So I am a total fangirl of Terry Gross. She interviews basically famous people with interesting stories, and she actually has a segment from like 2 weeks ago where she interviews a psychologist about a book she wrote about the science behind addiction. And this person also actually happens to be here we go. All right, Sydney. Sorry, you missed my personal anecdote. That's okay. All right. Okay, so carbohydrates. So whenever we're going to be doing a lecture-focused activity, I'm going to start off by actually flashing the learning objectives for you. I can copy and paste them and put them in a Word document so they're with the others for continuity. But just out of habit, when I do lecture style, I usually put the learning objectives at the front. So these are in your mind while we're going through the lecture, but I'm going to save you the boringness of reading through them individually. All right, so what are we going to talk about today? We're going to talk about the basic chemistry of carbohydrates. They're linear chains that form rings in solution. We're going to look at individual sugars. We're also going to see what they look like when they join up to form polymers. We're going to see that carbohydrates can form complexes with proteins. And then we're going to talk about some of the important roles that carbohydrates play with energy storage, with structure, and also with cellular localization and recognition. So carbohydrates, like proteins, are pretty cool. They can actually work with proteins to get jobs done. But one really quick distinction that I want to make about carbohydrates that makes them different from proteins, which we have studied, and nucleic acids, which we haven't quite gotten to, is that carbohydrates are not template-based. So does anybody remember what I mean by it's not template-based? Yes, no. Exactly. So there's a specific rhyme and reason to which the individual monomeric units are put and protein smaller. The DNA is put in a order that codes for RNA, and then codes for a particular type of protein. And of course, we've beaten into the ground with a rusty old stick that the sequence of the amino acids matter for the folding, the structure, and the folding, and therefore the binding and enzymatic activity, if applicable, of different proteins. Carbohydrates, not so much. They string together, but we're not so much worried about that they form a particular structure and do a particular job, as that they're arranged in such a way that we can easily access their individual monomers to then use for fuel, or they're kind of a big glob that's acting as structure. And I'm overgeneralizing a little bit, but the point is that the sequence, there really isn't a sequence thing going on here. It's more of just random linkage. And you'll see what I mean in a second. So carbohydrates are super, super abundant. They're really common in nature. And there's a couple ways that we can classify their structure. The first is whether or not, or essentially they're polyhydroxy. And so polyhydroxy means that every carbon has some kind of an oxygen containing functional group coming off it, whether or not it's an alcohol, a ketone, or an aldehyde. Usually polyhydroxy, hydroxy meaning OH. And they're either polyhydroxy aldehydes or ketones. And so an aldehyde, if we remember from organic chemistry, means a C double bonded O bonded to an H, or a keto, or an aldehyde or a ketone. So the ketone means, again, we have a C double bonded O that's bound to two other carbons. So the double bonded oxygen is going to be contained within the chain for a keto carbohydrate, and it's going to be on the end for an aldehyde carbohydrate. The chemical structure for these molecules typically is CH2O with N for the number of carbons that are in the chain, and the two words that we use to refer to these different classes of sugars are aldoses and ketoses. So which one of these guys do you think is the aldose? The one on the left or the one on the right? Right, and it's their aldotriose. The one on the left is going to be an aldose because it has an aldehyde, whereas the one on the right is going to be a ketose because it has a keto group. But no matter what, to be a carbohydrate, you're going to see that there's going to be one carbon that's going to have a double bonded O bound to it, and all the other ones are going to have hydroxy or OH groups or alcohol groups on them. Now, we also can classify the sugars based on the numbers of carbons they have. So we can see here that these are trioses, which means they have 3 carbons. The smallest sugars that we're going to see are going to have 3 carbons. We can also characterize sugars based on the number of individual monomeric units. And so we refer to an individual sugar within the context of a polymer as a saccharide. So a single sugar is a monosaccharide. A disaccharide is 2 sugars linked together. And so we'll see a lot of sugars where we have the basic unit being either a mono or a disaccharide. Oligosaccharides are when you have multiple sugars linked together, so really more than 2. And then polysaccharides are the lower limit for calling something a polysaccharide is when you have 20 or more sugars linked together. Okay, so it turns out that just like with amino acids, and by extension to nucleic acids, which we haven't seen yet, but the stereochemistry of sugars matters. All right, and so if we take a look, just like with amino acids, there are 2 stereoisomers of sugars. You can have D sugars or you can have L sugars. And so it turns out that the convention for sugars is the opposite for that of amino acids. So for amino acids, most amino acids in nature have L stereochemistry. For sugars, D. If you think things couldn't get any more confusing. Sugars are opposites of amino acids. Okay, so there are going to be, I was going to say, you all are going to groan when you hear this, but just like with proteins and nucleic acids, oh, rats. Okay, just like with proteins and nucleic acids, there's going to be some element of memorization with sugars, and there are some sugars that I am going to want that I would like you to memorize, but I'm going to tell you explicitly which sugars those are. And so these four are ones that you need to memorize. Now, what I will say is you are going to be responsible for knowing the structure of nucleotides, but also nucleotides with the ribose sugar and the phosphate attached. So these ones, these sugars that you're going to be memorizing, these are sugars that you're going to see elsewhere throughout the semester. So just think of it as an investment into nucleic acids and glycolysis. So you're already going to be doing ribose and 2-deoxyribose for nucleic acids, and you're going to need to know glucose and fructose for glycolysis. You're just making your life in April easier, okay? So you need to know these. And many of the sugars that we're going to be talking about and the sugars that we think about sugars are either going to be hexoses or they're going to be 6 carbons long, so glucose and fructose, or they may be pentoses. And the pentoses that we'll see the most are going to be riboses and ribose derivatives. Okay, so one of the things that's going to be really important, it's going to help us orient ourselves for sugars, is how do we number the different different carbons on the sugar? Well, the convention for numbering sugars is that you start at the sugar that has the double bonded O, or at the end that is closest to the double bonded O. So if we look at D-mannose, so okay, it says and these, I really just want you to know glucose. Okay, we just need to know glucose. Okay, we just need to know glucose. All right, so we need to know. So if we start at the double bonded O and work our way down through the chain to the end that has a hydroxy group on it, that's 6. Now, okay, so you may be wondering, how do I know whether or not a sugar is D or L? Well, it's determined by, for hexoses, the number 5 sugar, and for pentoses, it would be, I think, the number 4 sugar, or the number 4 carbon. So what's going to be the carbon that's in 1 from the last carbon on the numbering scheme? Okay, and whether or not the hydroxyl group is on the right or on the left. So all of these sugars are hexoses, and they all have the hydroxyl on the right at carbon number 5, or the carbon that's 1 in from the last carbon. This means that they're D sugars. Okay? And if you were to look and we were to go through and take the Fisher projection and make it into a perspective formula, or have it make a molecular model of it, we would see that it would be, that would fall in a D instead of an L. Now, an L would have the OH on the left and the H on the right. And I know this is a little bit of a simplistic description here, much more simplistic than probably what you got in OChem when you talked about D and L. But for our purposes, this is how you can quick, in a pinch, figure out if it's D or L. Now, that's how you can determine if amino acids are carbohydrates are D or L. But there are going to be carbohydrates that are going to be identical chemically, but they're going to be different sugars because their OHs and their Hs fall in different places or on different sides of the carbon, which means that they're going to translate into being up or down on the sugar, and that's going to affect their chemistry. Okay? So we have a special name for sugars that differ only at 1 carbon, and those are called epimers. And so in this case, And this particular figure, they have the carbon at which there's one difference from glucose. So mannose is an epimer of D-glucose at 2 position. Galactose is an epimer of glucose at the 4 position. But galactose and mannose are not epimers of each other because they're different at 2 different carbons. And so you may be wondering, you may be wondering why the side of the carbon matters and why it makes a difference chemically. I personally prefer representing sugars in the linear format, and that's how I will recommend you draw your sugars, especially because it's going to make it easier once we get into glycolysis. But sugars, it turns out, aren't just linear, and you probably have an idea of this from when you took organic or when you just looked at a picture of a sugar. I don't know if people just look up pictures of sugars, but you probably think about sugars in ring form. And so you may be wondering, why is Dr. GB showing me a sugar as a line when I'm used to seeing sugars like rings? And that's because sugars exist in an equilibrium between their linear and their ring forms. And so you'll notice here that depending on the carbon, the hydroxyl groups are either oriented up or down, and that's determined by whether or not the hydroxyl group is on the right or the left in the linear form. So if we look at carbon 1, it's on the right, so that puts carbon 1 on the down position. Same with number 2, it's on the down position. And so whether or not the hydroxyls or the hydrogens are on the side of the sugar can affect whether or not it's recognized by a certain enzyme or how it's stored. So that's just why that matters and why we care. But moving on to the point that we can represent monosaccharides either as linear or as rings because they circularize. And so what happens with the circularization reaction is you have the oxygen, in this case for a hexose on the number 5 carbon, it's the carbon that's one in from the last carbon. It will make nucleophilic attack. Okay, we see the reaction over here. It will make a nucleophilic attack on the carbonyl carbon. Those electrons will then abstract a proton from solution, and then you end up with the ring structure either on the left or on the right, where that carbon double bonded oxygen turns into an alcohol, and then the oxygen that's on number 5 is going to be that oxygen that's then contained within the ring. I'll take a minute and let that sink in. So the carbonyl oxygen is going to become an alcohol instead of an aldehyde or a ketose. And then the number 5 oxygen, which also happens to be the anomeric carbon oxygen, is going to be locked into the ring because that's the one that's making a nucleophilic attack. If we were to erase this line, we double bonded O there, and then that would be an OH. But I'm going to go ahead and, no, we're not going to mess with the eraser. Okay, so 2 equal sets of equilibrium arrows here. And you may be wondering, why are there 2 sets of equilibrium arrows if those sugars look exactly the same? Well, it turns out that depending on the angle at which that oxygen makes a nucleophilic attack on the carbonyl carbon, or from what side it approaches, you can either get a sugar and an alpha or a beta conformation. So the alpha combination happens when we approach the angle, when we approach the carbon, trying to see how to make this work. When the carbon approaches the carbon and such that the way that the new 1' OH is going to be down. And then the beta is when the resulting, so we'll say, and the beta is when the new hydroxyl is up. So in the alpha formation, the new 1' hydroxyl is going to be down. And in the beta, the newly formed 1' hydroxyl is going to be up. And this is going to come into play when we start linking monosaccharides together to make 5-tri-oligo and polysaccharides. But for now, I know it's a lot of convention. But it's important to remember that sugars, you can represent them either way, but they are in an equilibrium between the two. I know I've done really hard work at trying to fast talk equilibrium to you guys. This is a case where we will see sugars in an equilibrium between the open and the closed form in the body. All right, so these are examples of what ring-closed sugars look like. So these are fancy names that we can use, the full formal names of these sugars. So glucose is actually glucopyranose in the long form, and that comes from the fact that the basis of glucanose is a pyran ring. And the same for fructose. The basis of fructose, it's a six-membered carbon or 6-carbon ring, but because fructose is a ketohexose, it's going to be a 5-membered ring. A 6-carbon chain, but a 5-membered ring, and that extra carbon, you're going to have an extra carbon hanging out on the off of the one position. It's important to remember. It's really tempting to call fructose a pento or a pentose, but it is in fact a hexose. It just looks like a pentose, and we often forget about that extra carbon. Okay, so sugars are really diverse. You don't have to memorize this table of sugars. But what I do want you to know is that sugars can be modified in a whole bunch of different ways. They can get amino acids put on them. They can get amines put on them to be amino sugars. They can have their hydroxyl groups removed to become deoxy sugars. So the classic example that we'll see of that is we have ribonucleic acid or RNA, which has a ribose ring, and deoxyribonucleic acid, where the ribose group is short 1-hydroxy group compared to the regular ribonucleic acids. And then we'll also see some weird things going on where we have acids jumped on, we'll have carboxylates, all kinds of weird things can happen to sugars. But for us, in basic biochemistry, we're mostly concerned about glucose, fructose, ribose, but this is a thing, depending on your interest, you may learn more about this later, but we're not going to dive into any more detail than a cursory glance. Now, the next topic we need to talk about are reducing sugars. And so what I mean by reducing sugar is if you take a sugar, remember, in order for this to occur, remember, we have to have a sugar that is in an equilibrium between its ring and its open form. So let's say we have glucose. Glucose is opening and shutting, opening and shutting. And it turns out that when glucose is opened, it can actually be oxidized by a metal ion. So it goes from being C double bonded OH, R, to R, C double bonded O, O minus. So because that carbon now has going from 2 oxygens and a hydrogen and an R to 2 oxygens and another oxygen, so 3, it's becoming oxidized, and so it's transferring it can transfer electrons to an oxidizing agent like copper 2+. So it turns out that carbohydrates that are able to open up can reduce metal ions and other oxidation agents. And so, but we can also use this to our advantage. It's actually used sometimes in enzymatic assays, and that we can use this property of open sugars, sugars to identify sugars, but also to quantify sugar production in very special cases. Okay, so the key thing is, again, in order for the sugar to be oxidized, it has to be able to linearize. And in order for it to linearize, You can't have that carbon involved in a glycosidic bond. By the way, I can't remember. We haven't gotten there yet. Okay. All right. Sorry for the craziness where I've been flashing slides. Okay. So let's say, for instance, that we have 2 molecules of glucose. So you have 2 glucoses. Now, one thing I will say is, so this is the carbon where the chemistry happens. That's the carbon that's attached to the oxygen that does the nucleophilic attack to close up the sugar. We have a special name for this carbon, by the way, and I can't remember if it was written down on a previous slide, but we call this the anomeric carbon. So the carbon at which the sugar opens and closes and does chemistry is called the anomeric carbon. It also happens to be the carbon that determines whether or not the sugar is D or L. Fun fact. Okay? And so I think it is, yeah, I think it is. Yes, it is. No, it's not. I can't remember. Don't quote me on that. Okay, we'll go back and we'll work through that. Either way, that carbon, the anomeric carbon, actually, I said that wrong, guys. The anomeric carbon is carbon number 1. So if we were to go through, I'm sorry, guys, I had a brain fart, and now the program is yelling at me for having a brain fart by protesting and stopping sharing. We'll start our sharing again. So the carbon, the carbon that's at the beginning of the ring. So this is carbon 1, the anomeric carbon, 2, 3, 4, and 5, and 6. So this is the carbon that's attached to the OH that does the chemistry. And then that's the carbon that has the oxygen where the double bonded O pops open to become a hydroxyl. And it's the carbon that's at the top of the chain that gets attacked. That is the anomeric carbon. And so that's going to be the spot that determines whether or not a sugar is reducing, is whether or not that spot is opening and closing. So whether or not we can do chemistry at that anomeric carbon, carbon number 1, whether or not carbon number 1 can open back up and become a double bonded O again, is going to determine whether or not a sugar is reducing. Okay, so my alarm went off, so that means I need to give you a 3-minute stretch break. I should resume that. Is determined by which side the oxygen and the hydrogen are on. And if that oxygen is making an attack, that side at which that oxygen is on, because of its stereochemistry, is going to influence the resulting structure of and therefore the stereochemistry of the sugar as a whole. Yes. 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 It's always the hydroxyl group on the fifth carbon. Yeah. It's always going to be the one on the fifth, because you don't want the one on the end, and you're going to have carbon number 6 hanging off of the ring. It's just whether or not you also have carbon number 1 hanging off of the ring too. Yeah. I wouldn't say that. You should know what makes it D or L. You should know that the left or the right translates into D or L. I would be more concerned that you are able to identify the difference between D and L. Yes. What's that? Okay. So yeah, so I would say that for the first question on the quiz, you're going to want to have the substrate. You're going to want to set up this. Or let me look. Do we have the quiz? Okay, let me look at a printout of the quiz. I could pull it up in my great. Draw the active site of papain. Yeah, I would say that to draw the active site of papain, I'm most concerned about the amino acid, but I feel like you'd also want to draw the substrate. Yes. Yeah. And for those of our listeners at home who missed this, so the anomeric carbon is the carbon that is going to be receiving the nucleophilic attack from the 5' hydroxyl. The 5' hydroxyl, or the second from the last, is always going to be the one that's doing the nucleophilic attack. But whether or not the anomeric carbon is carbon number 1 or carbon number 2 is going to depend on whether or not the molecule is a ketose or a hexose, or not a ketose or a hexose, a 5-membered ring or 6. I mean, gosh darn it! It's going to determine whether or not the double bonded O is an aldose, so it's on the end forming an aldehyde, or a ketose, meaning it's on the inside, so it's on the second carbon instead of the first. So this one, again, if we were to look at the linear form of fructose, fructose has its double bonded oxygen on number 2, so we're going to have an extra, a 5-membered ring and an extra hydroxyl and carbon hanging off, so carbon number 1 is there, whereas for glucose, our anomeric carbon is going to be at number 1 because it's an aldohexose, which means the last carbon is going to be attacked because that's what the double bonded oxygen is. Okay. All right. I was going to say, it can be confusing at first to wrap your head around, but once you get it, it's something that's fairly logical. Okay. So I said, okay, so we go here. So we have the 2 saccharides, we have the reducing ends. So chemistry, oftentimes it makes sense that chemistry might be done at the reducing end of a sugar, at least for one sugar, because that's the reactive part. And so in this case, to make the sugar maltose, we're going to have 2 glucoses come together. So when they come together, that's a hydrolysis reaction, or not a hydrolysis. When they come together, that's a condensation reaction, meaning they release water. And when they break apart, it's a hydrolysis reaction, meaning that water is added across that bond to make 2 separate sugars. And the OH goes there, the H goes here, etc. But if we take a look at maltose, you'll notice that the anomeric carbon of glucose number 1 is locked up, but it's connected to carbon number 4 of the second glucose. So in this case, I want you to take a look. Oh, well, I guess, okay, I was going to have you do it, but I didn't alter the slide to accommodate this. Maltose is a reducing sugar. Why is maltose a reducing sugar? If this anomeric carbon is locked up in a glycosidic bond, by the way, the bond between 2 sugars is referred to as a glycosidic bond. If there's a glycosidic bond locking up the anomeric carbon, carbon number 1 of the first glucose monomer, why is this a reducing sugar? You can take a minute, talk to your groups. You can actively ignore your group, whatever you want. I'm not going to enforce group work today, but it might help. Why? Yeah. I thought it could have like Does anybody have any idea? Okay. Okay. Did any group come up with an idea that they're willing to share? Wow, we're still a little uncomfortable. Okay, so does anybody remember what the definition of a reducing sugar is? What makes something a reducing sugar? Okay, so yes, it can linearize. So it can open and close. So it can open and close, and a result of it being able to open up, it can donate electrons to another molecule. So a molecule like copper. Okay, so let's take a minute. Okay, so as a group, why don't you talk about why the carbon I've circled, so the carbon number 1 in the first sugar, why can that not linearize? And what's different about the anomeric carbon on the second glucose? Let's answer those two questions. Why can't the anomeric carbon, carbon number 1, on the first glucose linearize? But what's different about the alpha carbon on the other glucose? Okay, take a minute, group up. Yes. So
between the glycosidic bond and the O-glycosidic?
they're the same. They're the same. Yeah. It's just one has the O in it and one forgot to put that it was an O-glycosidic bond. Pardon? Yeah, what's the difference between the two anomeric carbons? Yeah. Okay, does one of my groups want to tell me what's the difference between the two anomeric carbons? I heard some good words that I liked. Yes, Caroline. Okay, and then how about the other anomeric? So, okay, so you're going in the right direction, and that you're saying that the carbon on the right hand glucose can occupy either alpha or beta, but the one on the left can only occupy one. Can somebody else dive a little deeper and say perhaps why that is the case? That's part of it. That's just an observation, but we can dig down a little bit deeper than that. Yes. Yes, Ed. Mm-hmm. Exactly. Yes. So the reason why the one on the left is locked into one alpha or beta and the one on the right can go either is because the one on the left is locked into an O-glycosidic bond. So that carbon is, I guess, somewhat permanently donated, used its oxygen to be bound to a carbon on another molecule. All right? So that can't be alpha or beta. It's restricted because it's bound to another sugar. But the one on the right can still open up and close because it's not involved in a glycosidic bond with another sugar. So you can think another way of thinking about whether or not is something reducing or not is whether or not the anomeric carbon is involved in a glycosidic bond. And you're going to have sugars where you're going to have multiple monosaccharides bound together. Not all of the sugars are going to be able to reduce because they're locked into glycosidic bonds, but on the end probably will. And that is going to be really important for how our body works with and processes carbohydrates. Okay, so kind of getting at what Caroline's group was saying, Disaccharides or glycosidic bonds between two sugars can either be in the alpha or the beta conformation, depending on what the orientation is of the oxygen on the anomeric carbon that is involved in the linkage. So we can either have a beta or an alpha form, and certain sugars are going to have one of the other. So lactose is an example where we have galactose and glucose linked together through, this is the beta form of the linkage. Okay, and so we often draw a beta linkage as a swoop because this oxygen is up and this oxygen is down before they link together. Now, sucrose, this is the last sugar that you're going to be responsible for knowing. That's sucrose. Sucrose has an alpha linkage, and that's because both the beta and the beta. Whoops. This is a beta, or it's an alpha. Okay, so those are different sugars on different sides. So it's because the sugar for the hydroxyl for the beta or for the fructose is in the alpha position, and the sucrose for or the hydroxyl for the fructose or the glucose. Goodness, I'm sorry guys, I'm really tearing it today. This one is in the beta position. So the alpha linkage is between the alpha and the beta of another. So we can draw this linkage, often draw an alpha linkage as a U. So we think of the beta linkage as a swoop and the alpha linkage, oh goodness, that's not true. Apologies. In this particular case, the alpha linkage is, but it really just depends on whether or not the alpha or beta of the opposite sides. So in this case, we're both in beta. In this case, we're in alpha and beta, so it's a U. And then in trehalose, we see alpha and alpha, so it's a swoop again, just in the opposite direction. I'm not so concerned about you being able to identify the alpha and the differences between the alpha and the beta, especially since I just butchered it just a little bit. What I am concerned about you knowing is that the orientation of the alpha and the beta on either side can determine how that glycosidic bond comes together in space. Yes, Sydney? I actually don't know, but I would imagine that in this case, it would be showing the predominant form. Yeah, that it would be showing the predominant. I would imagine that sterically one of them would be favored over the other. And if we look back at the alpha and the beta slide, it shows the percentages that for glucose, the beta is favored over the alpha. So, and again, this is an equilibrium, but only a very small percentage is going to be linearized at any given point in time. Most of the time, it's going to be circularized. But yeah, and usually one alpha or beta is favored over the other. But again, I just really want you to know, so you'll have to know sucrose, but other than sucrose, generally speaking, and sucrose is an alpha to beta linkage. You just need to know that the orientation, the alpha and beta on the two ends of the bonds influences how you draw the bond and what the nature of the linkage is. Okay, so sugar. When you hear sugar, you associate sugar with sweet things. So you may be wondering, how do we detect sweet? Turns out that we have taste receptors on our tongue, and this receptor is dimeric. And so sugar molecules will go and bind to both subunits. There's a conformational change in that receptor protein that is then propagated on the other side of the cell, and it sends off a signaling cascade in our cells and in our brains that tells us something is sweet. There's a huge market for artificial sweeteners or non-caloric sweeteners such as aspartame and NutraSweet /NutraSweet, and these are molecules that only bind to one half of the receptor. They can't bind to both, so that's one difference. They also although they give off a sweet signal, they aren't processed like normal sugar. So our body doesn't recognize that they can be broken down and used for metabolism. In theory, it passes right through our body. And so how these sweeteners are able to our taste buds without triggering the metabolism cascade is the fact that if you have a molecule, if we look at the zoom in of the receptor, there's two pockets. And so X means hydrophobic, B means there's going to be a negative charge that those protons are going to interact with, and that means there's going to be basic or charged groups on the molecule that will interact with protonated groups in the protein. There's two pockets, and aspartame, when it's in the right stereochemistry, can bind to both pockets, which sends that sweet signal, but it doesn't bind the whole protein. It also doesn't look like sugar, so it won't get processed. Whereas if the stereochemistry were off on aspartame, that hydrophobic ring wouldn't be able to swing into that pocket, and then you would get a bitter signal instead of a sweet signal. That's more just kind of a fun fact. Again, the stereochemistry matters, and these artificial sweeteners partially, but don't totally bind the receptor. And that's part of the reason why they're able to trick our body. Now, I prepared this slide. I'm probably going to zoom over it a little bit for interest of time, but there's a bit of controversy about aspartame and whether or not it causes cancer. And just because there was a study out there that found a correlation between a certain type of blood cancer in rats and aspartame dosage, and they saw statistically significant tumor growth at half of the recommended safe level of aspartame for adults. And this really scared a lot of people. This was out in 2006, and I remember one of the immediate results was that Diet Coke started switching over from aspartame to Splenda, and it was a big thing. Well, studies that have come after that haven't been able to replicate these results. And even if they did see things happening with tumors, it was only in males, not females. And they weren't able to find a dosage-dependent relationship. So I wanted to use this as a moment to point out there's a lot of claims out there. There's a lot of studies. There's a lot of cutting-edge research about whether or not certain chemicals are okay or not. And so you as scientists and having access to the Wake Forest internet have the ability to look up scientific studies. So if somebody, talks about a claim or you see something on the news like, oh no, LaCroix is bad for you, you can go through and do a little digging yourself. And things to just remember are to check the source. Does it test humans? What's the size of the population? Does it test males and females? Or do they throw women out because their hormonal irregularities make them hard to look at? These are things to think about. Or is it in rats and mice? Or is it even just in cells? There are a lot of people out there that make claims based off of studies that are only done in cell lines. And anyone who spent any time with someone who's doing drug development knows that something works in a cell doesn't necessarily work in a rat and a human, let alone a rat. So keep that in mind. And also, is it a first study or are there other studies in a body of work that support this claim? And in this case, there really isn't a body of work to support the fact that aspartame directly causes cancer. So again, these are things to keep in mind. I think that these studies, when they come out, they should alert your attention, but don't have a panic attack. But then another thing that I wanted to point out is I wanted to point out another artificial sweetener. We've already seen aspartame. We talked about peptides, and aspartame doesn't get processed largely because, well, it's a peptide. It's a modified peptide, not a sugar ring. But what happens with Splenda? So Splenda, by the way, fun fact, even if aspartame doesn't cause cancer, you can get too much. And my mother-in-law actually did get aspartame poisoning. So if you drink a whole 12-pack of Diet Coke in a day consistently, it can happen. So don't recommend doing that. But still, give a Diet Coke once in a while. Okay, so Splenda is sucralose. What exactly is sucralose? So here's the chemical structure of sucrose. Here's the chemical structure of sucralose. So the three hydrogens that are oxygen hydrogens that have been circled have been replaced with chlorine groups. And there's something about adding those chlorine groups that enables sucrose to bind to the receptor in such a way that it sends off a sweet signal to our brain, but it doesn't get processed and sent through glycolysis or put into our glycogen stores. And this is playing a little bit into the what do we believe, what is out there, do we have enough evidence to support this? But there are several studies out there that have shown that sucralose alters the gut microbiome of rats and mice. So I wasn't able to find any studies yet that do humans, but there is a growing body of work that shows that sucralose to varying and other artificial sweeteners to other degrees actually can alter the populations of bacteria that live in our gut. So again, don't know that about humans, but to me, I don't know about you, my grandmother was diabetic. She used to complain about GI stuff. Perhaps this could have been part of it. I have no idea. But this is something that's out there, and I have a feeling we're going to be learning a lot more about the relationship between artificial sweeteners and our gut microbiome, as well as how the bacteria that populate our gut relate to our overall health in the next 5, 10, 15 years. I think that's going to be a really big thing. So anyway, if you look at these graphs, you can see that if you look at the control and the mice that have sucralose, those populations of some of those bacteria are going to change. So, yeah, but again, just in rats for now. Who knows what we'll see in humans? Now, we talked about monosaccharides. We've looked at some disaccharides. We saw the disaccharide you need to memorize, and now we know how to figure out which sugars, if any, within a multi-sugar unit are reducing sugars and those that are not. So, but as I mentioned, carbohydrates are really important for structure and also energy storage. And so particularly, we like to store sugars in polysaccharides or glycans. And the most common forms of monosaccharides that we use as fuels are starch for plants, glycogen for us, for animals. They also form the structural elements, so cellulose for plants, and chitin in animal exoskeletons. So they're really important, and you can either have homopolysaccharides, so glucose polymers, like we're going to talk a lot about. They can be branched or they can be unbranched, or we can have heteropolysaccharides, which have different sugars that are linked together to form either a branched or an unbranched polymer. So we're going to focus a lot on homopolysaccharides, specifically the ones we mentioned on this slide. So starch, this is the polysaccharide that's used for fuel in plants, and it consists of two components. And again, these are homopolymers, so it's all glucose. So we have amylose, which is insoluble in water, and it contains about 20% of the starch. And for amylose, it has an alpha 1-4 glycoside polymer. So it's literally just a chain, an unbranched chain of glucoses with 1-4 linkages. So like that linkage we saw, we looked at whether or not the disaccharide was reducing or non-reducing, that kind of a linkage, the anomeric carbon of the first one to the 4 carbon of the next glucose. So that's amylose. It's just a straight line, and we can see that there in the blue. Amylopectin, however, has an additional linkage. So amylose is not branched. Amylopectin is branched. It also happens to be soluble in water. We'll learn a little bit about that in a minute. And it actually composes the majority, so 80% of the starch. And so, like amylopectin, it has 1-4 glycosidic bonds. What also happens is that some of these molecules, so if we have 1-4, 1-4, 1-4, whatever, some of these are also going to be making 1-6 branches. So if we have the 6 up here, it'll be branching with the 1 of another molecule. So we can see here, if this is the main chain of our carbohydrate, so there's the glucose number 1. If we have glucose number 2, it's involved in a 1-4 linkage, and then its 1 is involved in another 1-4 linkage over here. The 6 carbon, the hydroxyl on the 6 carbon, is then going to make a glycosidic bond with the 1 or the anomeric carbon of another glucose, and then it's going to carry off in the other direction. It's a lot. You can sit, mentally play around, and manipulate it, but you're going to have to trust me that starch is a combination. It's mostly this branched amylopectin, but periodically you'll have some amylose that's just a linear chain wound in with it. So that's starch. The analogous structure in humans is glycogen. And we have a lot of glycogen hanging out in our muscles, as we're going to learn when we talk about metabolism. And so what happens with glycogen is highly branched. So it contains up to 1 unit of glycogen can contain up to 100,000 glucose units. And it's again, like amylopectin, it's 1 to 4, but with 1 to 6 branches. And so the overall global structure is it has like a center, and then the branches kind of wind out upon each other. And so it has like this, I like to think of it as like a spirally structure. And what this enables is you have is all around in one glycogen unit, you have a whole lot of ends that have glucoses with reducing sugars. And having all of these readily available ends with reducing glucose sugars hanging off the end means the enzymes in our like muscles that need to get glucose quickly to fuel things like jumping up and down like a crazy person can go and snap those glucoses off and then funnel them really quick into glycolysis for quick bursts of energy when we need it. So that's why glycogen has this structure, because our body needs to quickly access those glucose molecules for metabolism. Again, it'll make more sense in about 4 weeks. And unlike starch, glycogen is relatively compact. Now, we've talked about alpha and beta linked glucose polymers. So again, we've mentioned that whether or not those carbons in the bond are in the alpha or the beta formation can determine the conformation of that glycosidic bond. It turns out the glycosidic bond, specifically whether or not the first carbon is in the alpha or the beta, can have really big ramifications for the structure and even the solubility of those polymers. So if we have alpha linked, it turns out that starch is alpha linked. So if this is sugar 1 and that's sugar 2, this guy is going to be in the alpha, okay? And so it turns out that alpha-linked glucose polymers have an open curve conformation. And so because they have this open structure, it turns out that they are able to dissolve in solution, which is good for storage, and it makes them more appropriate. They can dissolve, and then the monomers can be relatively plucked off. The multiple ends make it much easier, again, for enzymes to come in and access it. And one of the enzymes that is responsible for this is alpha amylase. And we can find alpha amylase particularly in our saliva, where when you have glucose polymers coming in from food, we can digest it down into monomers, and those monomers can get added onto the glycogen so they can then get taken off later. The other type of polymers are beta-linked glucose polymers. And so again, a beta is when we have when the sugars are linked at their beta positions. Now, this is where we really see why alpha and beta matter in terms of that glycosidic bond. Because with beta-linked glucose polymers, when you have those sugars in the beta conformation, the hydroxyls on the glucose line up so they can hydrogen bond with each other. And so if you were to look at a ball and stick model of a beta-linked glucose polymer, every single group that can hydrogen bond is hydrogen bonding with another group within the polymer. So if we're looking at alpha-linked, those groups can hydrogen bond with water. And so they dissolve, because when you interact with water, you can dissolve. Same thing with solvating charges, and why we have polar and charged groups on the outside of proteins. Same thing with carbohydrates. Beta-linked, not so much. That's all taken up, all those hydrogen bonds that appear to be going off into space, that's not with water, that's with other carbohydrate groups. And so because of this, the shape is going to be different, and so is the solubility. So it turns out that beta-linked glucose polymers are fibrous, or they're insoluble because of that high degree of hydrogen bonding, and because they're so tightly linked together, it's really hard to get in there and chop up individual glucoses. Which, if you're using it for energy, like glycogen, that's awesome. But this is really good for structural things, or for plants. So we can't digest beta-linked glucose homopolymers, but turns out that termites can. So we can't digest beta homopolymers like cellulose, but plant termites and some other termites can. And again, cellulose, because of this, can also be really good for fuel. But again, beta-linked, not great for eating, good for structure, good for termites. And again, carbohydrates can form complexes with proteins. The names of these complexes come from the degree to which the complex is protein or whether it's carbohydrate. So peptidoglycans are where you have a glycan chain that's linked to peptides. This is the basis of a bacterial cell wall and is really important. This is how we take advantage of some antibiotics by messing up that cell wall of the bacteria. Proteoglycans make up the extracellular matrix. And so again, they're slightly different from peptidoglycans or glycoproteins and the ratio of carbohydrate to protein. But these guys are really important for maintaining the extracellular matrix, or the space between the cells in our body. And then the last type of protein carbohydrate complex, which I'm partial to because I like proteins, are glycoproteins. And so glycoproteins are proteins that have carbohydrates attached for some reason. You have two types, N-linked and O-linked. Attaching the carbohydrates affects the charge, size, etc., all these different protein properties that we think about when we're trying to purify them. And they're often important for extracellular recognition. And it can affect the function of the protein. You need to know that there are N-linked or O-linked proteins, or glycoproteins, and you need to know that O-linked carbohydrates can either happen at a serine or a threonine, whereas N-linked can only happen as asparagine. So all of you who wanted to do chemistry with asparagine, this is where asparagine has its shining role. It is really important for glycoprotein function. And again, these are always going to be attached to the anomeric carbon of the sugar. So when you link carbohydrates together to make like starch, sometimes you can have them going in other places. But if you're going to do chemistry with like an amino acid, it's going to happen at the anomeric carbon. Sugar, so sugars are really important when they're, whether if they're a substrate, we'll see this with nucleic acids, or if they're attached to a glycoprotein, they can be really important for sugar-protein interactions. So again, we saw how the arrangement of the alpha and beta could determine the orientation of the hydroxyls and the hydrogens, and therefore the hydrogen bonding, whether or not the polymer hydrogen bonded with itself or water. Similarly, sugars can have a hydrophobic side and a hydrophilic side, and that can determine whether or not a carbohydrate can interact with a protein as a substrate, or whether like a polymer can get chopped up. But it's really important, this comes really into play for cell recognition and signaling, also adhesion, which we're not going to talk a whole lot about. And again, glycoproteins hang out on the surface of the cell, and they could be really important for again, for signaling. So we see it with viral and bacterial recognition, with wound healing, connective tissue maintenance. We see it sometimes with vaccines, but also these glycoproteins on the surface of cells are actually what determine blood group antigens. So that's one way that affects all of us, whether or not we're different blood types, is whether or not we have certain sugars on the outsides of our cells. And with that, we have made it through carbohydrates. All right, so the assignment for nucleic acids and nucleotides has been posted to the Sakai site. It's not very extensive, but there is a video. There's a short pre-assignment, and then also I have recommended reading for 8.1 and then part of 8.2. Again, quizzes due beginning of class. If you have questions about enzyme kinetics or similar problems, so perhaps if you worked the exam I'm happy to answer those questions, but I will not answer questions specifically about the content of the quiz. And for this class recorders, you don't have to send pictures for this. Yeah, this is fine. We didn't do a whole lot of formal business on the way with that one.